enzyme is methylase enzyme. Methylase enzyme, which before the activity of REN, before the activity of restriction endonuclease, methylase enzyme adds many methyl groups to the bacterial DNA. So due to the addition of methyl groups, REN doesn't harm the bacterial DNA. Viral DNA is without methylation, so that's why REN directly attacks on viral DNA and destroys viral DNA and makes viral DNA into fragments. So this process is called restriction modification system. So restriction endonucleases are releasing in bacteria for defense purpose. That is important. Defense purpose. Right? So restriction modification system we studied yesterday. Now, so today we have to study about tools of recombinant DNA. Tools of recombinant DNA technology or rDNA technology. So what are the tools we are going to use in a recombinant DNA technology or genetic engineering, right? So here tools mainly there are three types of tools. Three types of tools we are going to use. Number one, enzymes. Right? So different types of enzymes are needed for rDNA technology. Second, cloning vector, or generally we can call vector, right? Vector or a cloning vector. So third one is competent host. Right? Competent means accepting. See, we studied yesterday the basic steps in rDNA technology. First of all, we have to maintain, we have to create a recombinant DNA. That recombinant DNA is introduced into a host. Right? So that host is called competent. Competency means accepting. So after making recombinant DNA or rDNA, that should accepted by the host. Then only our experiment completes. Right? So then only the experiment will be meaningful. Right? So that's why here major tools are three types: enzymes, cloning vector, and a competent host. These three are the main tools we have to use. Now, the first part, enzymes. What kind of enzymes we are going to use in a recombinant DNA technology? So here also we are using three types of enzymes we need. Three types of enzymes are needed. Okay? Number one, they are cleavage enzymes. Cleavage means cutting. So cutting enzymes. We know that DNA has to be cut into fragments for isolation of our desired gene. So to get desired gene, we have to cut DNA into fragments. So those belongs to this category, cleavage enzymes. Second kind of enzymes are DNA ligase. Right? DNA ligase. Ligation means union. Right? DNA ligase is the second type of enzyme. Here I am writing the third one, that is a DNA polymerase. DNA polymerase. So cleavage enzymes, DNA ligases, and DNA polymerases. So these are the three main enzymes we are using in recombinant DNA technology. So now cleavage enzymes. So cleavage enzymes. Cleavage. So cleavage enzymes are very, very important enzymes. Right? Say all these. So they are used to, here I am writing, they cuts DNA into fragments. They cuts DNA into fragments. That is the use of cleavage enzymes. And here we know that all these are, I am writing only important points which are very, very essential for entrance examination. Right? So they cuts DNA into fragments. So they belongs to, all these are belongs to the major group or class, that is nuclease. Very important. Nucleases. Nuclease. Nuclease enzymes are the enzymes which cuts DNA, which cuts nucleic acids. So they were named as nucleases. Of course, we know that nucleases are of three types. Nucleases are three types. Three types of nucleases. What are they? Number one, exonucleases. Exonuclease. Right? Say second type is endonuclease. Endonuclease. And final third type is restriction endonucleases. Of course, we are simply calling them as REN. So it belongs to group nuclease. They are of three types: exonucleases, endonucleases, and restriction endonucleases. These three are having their own different functions. So according to their cutting nature, they were divided into three different groups. Okay? So here I am writing number one, that is exonuclease. Exonucleases. Right? Exonucleases simply they cuts DNA. They cuts DNA at ends or edges. What ends? 5 prime or 3 prime ends of DNA. So any gene located at the end of DNA that can be isolated by using exonuclease. Right? So simply here I can draw a simple DNA molecule here. It's a strand of DNA. Imagine it's a 3 prime, 5 prime, and this one is the complementary strand. Here I am writing. It's a complementary strand. So it is 5 prime, 3 prime, right? Oh. Then now I am drawing a fragment here, right, which is a 3 prime and 5 prime fragment. So this enzyme cuts DNA at this location. So only at edges, exonucleases cuts DNA at ends, edges of a complete molecule. Right? So they cuts DNA at the edges. Right? So they are exonuclease enzymes. Yeah? Then